All right, so welcome to Module 13, where we're going to get into Chapter 11. And Chapter 11 is focusing on the nervous system. So we're going to talk about all the different characteristics of the nervous system, the structures that make up the nervous system, and we'll actually learn how electrical impulses, which is what we refer to as action potentials, are established and propagated across the nervous system so that they can send signals to tell the rest of the body what to do. All right. So some general characteristics of the nervous system is one, that the nervous system receives information from several different sources and it's receiving this information simultaneously. So there's all kinds of stimuli and electrical signals being sent to and from the nervous system at any given time. And the job of the nervous system is to receive that information and then to integrate that information. So the nervous system basically has to process incoming information, compile it, determine necessary responses to that information, and then send out those responses. And this has to happen extremely fast for our bodies to work appropriately. Okay, sorry about that. I had to pause the video. My son was playing with his dinosaur. Okay, so nervous system will receive information, process, integrate that information, and it's doing this on an extremely fast time scale, right? So it's receiving it, integrating it, and responding to it in a fraction of a second, right? And our body has to be able to do this in order to make all of our organs function, work together, keep homeostasis, and have everything working appropriately. And the responses that it's generating are responses like muscle contraction. So whenever we walk or move, or it could be involuntary muscle contraction. So, stomach contractions, or the breathing responses for our respiratory system. It could tell glands to release specific hormones or other secretions. And it could also be conscious control or involuntary movement, right? And so the nervous system is really important for kind of bringing all of these things and making sure they all happen and work appropriately. Now, the nervous system can be divided into two kind of major parts. You have the central nervous system, or the CNS. Then you have the peripheral nervous system, which is the PNS. So the CNS, or the central nervous system, is made up of the brain and the spinal cord. And we'll talk about the central nervous system towards the end of this chapter. And the whole job of the central nervous system is to really receive information brought to it from the peripheral nervous system, to process that information, and then to transfer information. The PNS are all of the nerves in the body that exist outside of the central nervous system. The peripheral nervous system can be further subdivided into the sensory division and the motor division. The sensory division carries information to the central nervous system. The motor division carries information away from the central nervous system. And then the motor division can be divided even further into somatic and autonomic divisions. So somatic is basically your voluntary control, and your autonomic are the involuntary structures that we don't control, right? We have the involuntary control over. Now, this autonomic division can also be divided into what's known as the parasympathetic and the sympathetic divisions. Okay, we have a figure here that kind of shows that breakdown a little better. So you have your central nervous system here. Everything else in this box is the peripheral nervous system, and they're bringing information to the central nervous system and carrying information away from the central nervous system. Now, the sensory, remember, is the input. So any kind of signals from the external environment, from tendons, muscles, or internal organs will be taken to the central nervous system, processed, and then appropriate responses will be sent to the motor nervous system, where it will either be voluntary control or involuntary control. Okay, so somatic is the voluntary control, and autonomic are the involuntary control. Now, the autonomic can be broken down into parasympathetic and sympathetic divisions, which we'll talk about towards the end of the chapter. And these are basically part of the fight or flight responses. And so they're important for increasing heart rate or decreasing heart rate, or like your scared responses or your calm responses. And we'll talk about which one does which once we get towards the end of the chapter and talk specifically about those. Now, in all of these divisions, so in each of these divisions, whether it's the peripheral nervous system and its subdivisions or the central nervous system, all of these systems use neurons as communication. And so neurons are those specialized cells for communication in the nervous system, and they generate these electrical impulses that sends the information from neurons to neurons or neurons to target cells. And there are different types of neurons. So you have your sensory neurons and your motor neurons, and they have the exact same function as the sensory nervous system and the motor nervous system. So sensory neurons receive stimulus and transport it to the CNS. Motor neurons take information from the CNS and transport it to other target cells, tissues, muscles, organs, etc. Now, interneurons, they transmit information between various components of the central nervous system. So they're kind of these like connecting neurons. Now, if we look at the structure of a neuron, there are three main parts to a neuron. You have the cell body, the dendrites, and the axon. And we've briefly talked about these when we talked about the different types of cells several chapters ago. But we're just going to kind of go over this again just to make sure everybody's on the same page. And so the cell body is that main part of the cell, right? If you remember, neuron has like this circular structure. That's the cell body. It has a nucleus, right? And so that's the cell body. Okay? Has a nucleus, the cytoplasm, and all the other organelles. Now, that on that cell body, there are little projections that come off. And these bring information to the cell body and then carry information on a longer projection. These smaller projections are the dendrites. This longer projection is the axon. And so the neuron will receive information from the dendrite, carry it down the axon, and then it will then move to either another neuron or a muscle or whatever other the target structure is. So your dendrites are small, slender extensions that come off that cell body and receive incoming information. The axon is a singular longer extension that is specifically developed to conduct electrical impulses away from the cell body towards a target structure. Okay. So here's a figure that shows a couple of neurons kind of working together. So you have, say, a puncture from a splinter here. You have receptors that are picking up that stimulus that will carry it to a dendrite, that will then carry it down to the axon. Okay? And the cell body here, then we have the other axon being propagated down. And then that axon will transmit that information to another neuron. It will go across the dendrites, across the cell body, down the axon to a third neuron, right? That will then propagate it down to the muscle. Okay? And so in this example, we have all three of the different types of neurons here. You have your sensory neuron bringing information to the CNS, the interneuron carrying information through the CNS and bridging the gap from the sensory to the motor, where the response is carried through the motor and telling that muscle to, if you're stepping on a splinter, it's telling you to lift your foot up. If you rub your hand over a piece of wood, it's telling you to pull your hand back. So it's initiating those responses. So you have your sensory neuron, your motor neuron, and then your interneuron. Now, the way that this information is carried down the neuron is through an electrical impulse. This electrical impulse is what we refer to as an action potential. And the neurons generate and transmit these action potentials throughout our body. And the action potentials are a way for our cells to communicate across the nervous system and to communicate with other cells and muscles and tissues and organs in our body. And the way an action potential works revolves around the sodium-potassium pump, right? We've talked about this sodium-potassium pump a little bit, and we said that the sodium-potassium pump is to maintain a specific cell volume. And remember, if you have a cell with a sodium-potassium pump, you have 3 sodiums being carried out and 2 potassiums being carried in. And if you remember, that leads to a net positive charge on the outside and a net negative charge on the inside. So not only is the sodium-potassium pump useful for maintaining the salt concentrations for tonicity, but it's also very important because it's maintaining a electrical gradient across the cell membrane. So outside of the cell membrane, you have a positive environment. And inside the cell, you have a negative environment. Okay, so that's really important to remember. Now, the negative charge that exists on the inside of the cell is what we refer to as the resting potential. Okay, so this is the measurable difference in voltage due to the change in charges across the cell membrane in a cell that's resting. And generally, this is about minus 70 millivolts. Okay? So inside the cell, it's about minus 70 millivolts at a resting neuron, so a resting membrane potential. And so that basically means that the interior of the cell is negative relative to the outside of the cell. Okay? So this is basically what we have going on. We have the inside of an axon, which is this area here. Notice that has a negative charge. So this is minus 70 difference here. Okay? Because the outside is positive. Right? The outside area of the membrane outside of the cell is positive. And again, that's set up by that sodium potassium pump because 3 sodiums are pushed out, 2 sodiums are brought in. So that leaves a negative net negative charge and a net positive charge across the membrane. Now the difference here is about minus 70 millivolts. That's the resting potential. So now that we've established the resting potential inside of a neuron, right, or across that cell membrane, the plasma membrane of the neuron, we're now going to talk about deviations from that resting potential and kind of what causes that. And so if you have a stimulus, right, so you have some sort of external stimulus to stimulate a neuron, that leads to what's known as a graded potential. This graded potential leads to changes in the resting membrane potential. So remember we said that that's minus 70. Right? Now, depending on what the stimulus is, it could either depolarize the cell or the membrane, or it could hyperpolarize the membrane. Depolarization is the resting potential becoming positive. Hyperpolarization is becoming more negative. So if it depolarizes, it increases in membrane potential. So maybe it goes from minus 70 to, let's say, minus 10. If it's hyperpolarized, then maybe it goes from minus 70 to minus 90. Right? So it becomes more negative. Right? So that's hyper, and then positive is depolarized. Now we can get stimuli, every now and then, and it doesn't really cause anything to happen. It just causes like a slight change in the resting potential, and then the resting potential will go back to normal because that sodium potassium pump is always working, and then it will kind of fluctuate a little bit after that stimulus. But we can get either a strong stimulus or several stimuli in a row that leads to several graded potentials that occur relatively quickly, either in space or time. And so they're adding together. And so if you have like a graded potential, right, where we're starting at minus 70, and then you have a stimulus and it changes it to minus 60, and then all of a sudden before the potential can go back to resting, you have another stimulus and it gets to minus 50, and then another one to minus 40, all before it can go back to resting, right? So you get this large depolarization because of that. So they can add up and eventually reach what we call a threshold or a trigger point. And if that graded potential reaches that trigger point, then an action potential will follow, and that action potential will trigger a response or trigger that neuron to send a signal to the nervous system that something is happening, whether it's you need to move your muscle or release an enzyme or a hormone, etc. So here's a figure looking at kind of these different things that we just talked about. Right, so here's the resting membrane potential of a cell, minus 70. Everywhere you see an arrow is a stimulus. Okay? And here you see a depolarization, so becoming more positive. So this is a depolarizing graded potential. Here you see a hyperpolarizing graded potential, becomes more negative. Here is where you see your summation. So you have two stimuli right back to back. You get one graded potential, but before it can go back down, you get another stimuli that causes a summation eventually. So it causes the resting membrane potential to become much more positive. Okay. Now, in this example, minus 50 millivolts is our threshold, right? That dashed line is representing the threshold. So if at any time this resting membrane potential becomes depolarized enough to where it hits that threshold, an action potential will happen. Okay. And this, so from here to here, this is an action potential. And so an action potential is just this sudden reversal of the membrane voltage. So it basically becomes much more positive compared to its resting membrane potential. And so that action potential, remember, is initiated when it has a graded potential or a summation of graded potentials that cause that threshold to be reached. Depolarization is the upward movement in that action potential. So remember we said that action potential looks kind of like this. So this upward trend is depolarization, okay? So during that depolarization process, after the threshold, when that action potential is happening, voltage sensitive sodium channels will open. So these channels aren't open during resting because they're only sensitive to changes in the voltage across the membrane. So when that threshold triggers, that's causing those voltage sensitive sodium channels to open up. And because those sodium channels open, you get an influx of sodium inside the cell. And that's causing the voltage inside the cell to become much more positive than it is at resting membrane potential. So it reverses the voltage across the membrane and the interior actually becomes positive. Okay. After that happens, you get repolarization where the Na channels will close, but then potassium channels will open. And then those channels will allow potassium to move out of the axon. And this brings the interior of the axon back to its negative voltage and restores that resting membrane potential. However, as these sodium ions are moving out, the proteins don't close exactly when that resting membrane potential becomes minus 70. It actually becomes a slightly more negative, so around like minus 70, minus 80, and becomes temporarily hyperpolarized. And over time, the sodium potassium pump will reestablish that resting membrane potential of minus 70 millivolts. Okay. And so that normal activity will restore that normal resting potential. We have a figure here kind of showing this process. So here we have your threshold being met. This increase is depolarization. So when this threshold is triggered, this happens up here, depolarization. So your voltage sodium channels open up, sodium floods into the cell, bringing in lots of positive ions. And so the electrical charge of inside the cell becomes more positive. And then after that sodium floods in, your membrane potential is now around positive 30. Then the sodium channels will close, and then your potassium channels will open and allow potassium to leave the cell. So now you have a lot of positive ions leaving the cell. So since you have positive ions leaving, the cell becomes negative again. Okay. And then, like I mentioned, those potassium proteins, they don't close exactly when that resting or that membrane potential becomes minus 70. It actually gets slightly more negative. You can see that here. This is the hyperpolarization after repolarization. and then the reestablishment of that resting membrane potential will happen after the potassium channels close, and then the sodium potassium pump continues working, and it can kind of self-regulate itself back. Now, what this figure also shows, so not only does it show the action potential in green, so this whole process, this whole kind of structure graph is an action potential. What these smaller curves are showing is the movement of sodium, which is the orange line, and potassium, which is the green line. That's showing you the influx of each of those ions relative to the action potential. Okay. Now, a couple things we want to mention about the action potential is that it's an all or none process. When the threshold is met, an action potential happens. It either triggers or it doesn't trigger. So that's interesting too, because that means that the action potential is always the same speed and the same voltage for the same type of neuron. Okay, and we'll talk about when neurons can have different speeds and strength of action potentials, and that's based off of the size and the diameter of the neurons, and we'll talk about that a little bit. But the big thing here is that it's all or none, and once it's triggered, it's always the same speed and voltage in the similar neuron. It's also self-propagating, meaning that it continues to propagate itself in the next region of an axon, and it actually moves down like a wave. Okay, so let's say if you have an axon and you have sodium to flood in here, potassium to flood out, that's causing an action potential here. And then what that's going to do is cause another action potential here, right? So you get another, and then another, and then another one. And so action potential propagates down the axon. Right? So this is the axon. But this propagation is very, very fast, right? So 1/10 of a second, okay? And the number of action potentials per unit of time actually determines the strength of the stimulus, okay? So stronger stimuli will generate more action potential per unit of time, and so that gives you more action potentials. And the speed of the action potential, like I mentioned, is always the same for a specific neuron, but the speed is different between different neurons, because different neurons, depending on where they're located, so if they're in your brain or if they're in your arm or your spinal cord, will have different diameters in the axon. So maybe one axon is this big, another type is this big, etc. And so the larger the axon, the faster that can travel. So you can think of it as like electrical cords or drop cords, if you're familiar with that. The larger they are, the more electricity and the more efficient they are at propagating those electrical signals. The same thing for axons, right? Okay, so something that helps with the transmission of these action potentials and electrical signals are neuroglial cells. Neuroglial cells actually make up about 80% of nervous system cells. They have a couple of different functions. So they provide support, they provide protection, and they also help to provide nutrients to the cells. So the glial cells do not transmit the action potentials, And we'll talk specifically about what they do. But there are two different types of neuroglial cells. You have Schwann cells and oligodendrocytes, and depending on where you find the glial cells determines the type of glial cell that exists on the neuron. So, for example, the Schwann cells form the myelin sheaths in the peripheral nervous system, and the oligodendrocytes form the myelin sheaths in the central nervous system. So Schwann cells is the PNS, oligodendrocytes is the CNS. Now, the role of the myelin sheath, these neuroglial cells, one, it helps to save the neuron energy. Two, it speeds up the transmission of the impulses. It basically makes the transmission of these action potentials a lot more efficient, and it creates a specific type of conduction pattern, which is known as saltatory conduction. This is basically a leaping pattern, so something like this, right? So it kind of hops down the neuron. And I have an image in the next slide to help you kind of see that represented in figure form. And another function of the Schwann cells are to help damaged or severed axons regenerate. And so the roles are pretty similar from Schwann cells to oligodendrocytes. It's just that you find them in different areas of the nervous system. So one's the peripheral, the other one is the central. Now, this is what we mean whenever we talk about saltatory conduction. So instead of the action potential kind of moving down the neuron like this, it jumps, right, like that. And that is what we mean by the saltatory conduction. So these cells here are the glial cells. So it's the Schwann cells or your oligodendrocytes. And this is a layer of myelin, so what's known as the myelinated sheaths, that's insulating the neuron. Now, between these myelin sheaths, there are gaps, like we see here and there. These gaps are known as the nodes of Ranvier, and this is where the action potentials propagate. And so you have your action potential propagate at these nodes and are carried down the neuron. Okay. And so to finish up this video, we have a little animation, and then we're briefly going to talk about some disorders that are associated with the myelin sheaths. So actually, we'll just talk about these disorders first, and then we'll go back to the animation and finish off with that. And so there are some disorders associated with the degeneration of myelin sheath. One is MS, multiple sclerosis. The other is amyotrophic lateral sclerosis, or also known as ALS. And with MS, you basically get damage to the myelin sheaths in the brain and the spinal cord. And as you get damage to those myelin sheaths, you lose efficiency in how your action potentials are transmitted. And so you can get the feeling of weakness, visual impairment, because those signals aren't sent efficiently anymore, and basically forms of incontinence. Now, ALS is damage to the myelin sheaths in the motor area of the spinal cord. And so because you start losing kind of efficiency and transmission of these motor neurons in that area of the spinal cord, your muscles will start to weaken and waste away because you're losing that progressive kind of use of those muscles themselves. Okay. And so we'll look at this animation that kind of covers a lot of what we've mentioned already. And then that'll be it for this video. And then we'll kind of go into the transformation of information from a neuron to a target cell.
Neurons have three principal regions: a nucleated cell body, dendrites, and an axon. The cell body is the nutritional center of the neuron and contains densely staining areas of rough endoplasmic reticulum. Dendrites are thin, branched processes that extend from the cytoplasm of the cell body, providing a receptive area that collects and transmits electrical impulses to the cell body. The axon is usually a longer process that conducts impulses away from the cell body. Nerve impulses originate in the axon hillock, an expanded region where the cell body and axon meet. Axons can vary in length from a millimeter up to a meter in length. Most of the axons outside the brain and spinal cord, and some of those within, have an insulating outer layer called a myelin sheath. Outside of the brain and spinal cord, specialized glial cells known as Schwann cells form the neuron's myelin sheaths. A different kind of glial cell called an oligodendrocyte performs the same function in the CNS. The glial cells wrap the axon in layers or sheaths of tightly compacted plasma membrane, which forms the myelin. During development, the Schwann cells roll or wrap around the same spot on the axon, such that each new layer overlaps the previous ones. Each Schwann cell covers only about 1 millimeter of an axon, leaving gaps of axon exposed between adjacent myelinated segments. These gaps in the myelin sheath are known as the nodes of Ranvier, and their presence is very important to the speed at which a neuron transmits messages. The myelin sheath acts as an electrical insulator, allowing the nerve impulse to travel quickly and passively between the nodes. At the nodes of Ranvier, the impulses are actively regenerated. As a result, the nerve impulse jumps successively from one node of Ranvier to the next in a type of transmission called saltatory conduction, which is up to 100 times faster than signal conduction would be on an unmyelinated axon of the same diameter. Not surprisingly, the axons that are responsible for conducting signals over long distances, for example, spinal motor neurons connected to somatic muscle, are typically myelinated.
All right, so that does it for the first video on the nervous system. So like I mentioned, we'll pick back up where we talk about and discuss how information is transferred from a neuron to a target cell or another target neuron, and kind of how that process happens. Okay? And so with that, we'll see you in the next video.